Hi guys, welcome to my YouTube channel. I'm Dr. Suchita Verma and today we are going to start another topic, that is PCR. Now what does PCR stands for and what it is used for? PCR stands for polymerase chain reaction. So PCR, it was given by Carey B. Mullis and it stands for polymerase chain reaction. Alright, so now when we talk about PCR, the very first thing that is important is what is PCR and why do we do that? So if I simply talk about the principle of PCR, it says that it is the enzymatic amplification of a gene or a DNA, right? So what does we understand about amplification? Now most of the time amplification is used for volume, to increase the volume, to enhance the volume, but here amplification refers to the number of copies of a DNA. Here we are increasing the amount of DNA. The same DNA which is available in a very small amount is being amplified a number of times, so that we get multiple copies of DNA which can be used for further research work, for further studies, or maybe say for further research work to be used in near future. Right. So now PCR, as it is suggested, it is the amplification of DNA. Means we are going to make more number of copies of DNA, multiple copies of the same DNA or multiple copies of the same gene. Right? Why it is called amplification? Because we are increasing the number of DNA. Why are we saying that it is enzymatic? Because in this process a lot of enzymes are involved, right? So that is why with the help of the enzymes we are making the multiple copies. Hence the principle is the enzymatic amplification of DNA, right? Alright, now PCR, as the name suggests, that it is a chain reaction, means there will be n number of reactions one after the other so as to get the final product. Okay, now PCR, it involves 3 steps, but before talking about these steps, let me quickly give you an idea that why do we use PCR. So supposedly there had been a crime situation and now we are on a crime scene and there is a blood smear. So now we want to go for some test and we want to find out that who is the culprit. So we need to have the DNA sample. And that DNA sample is being obtained maybe from the blood smear or maybe from the hair that is there at the crime scene site. Alright, so now we can take that blood smear and we can isolate the DNA. Now as we know that RBCs, they don't have nucleus, platelets, they don't have nucleus. So there is a very little possibility of having, enough amount of DNA that is being used for the investigation purpose. What if I lose all the DNA from the investigation site when something goes wrong in my laboratory? I will be left with no DNA. In that case, what can I do? So to avoid such misfires, avoid such difficulties, such problems, there comes the role of PCR. We all know that we can isolate DNA from WBCs, but WBCs are very, very less in number. So here comes the role of PCR, that we can amplify the number of copies of this DNA so as to secure or to have more number of DNA which can be used in further investigation, or in case by mistake something goes wrong, something bad happens and we have lost the DNA, we still have the similar copy of this DNA with us so as to continue the work. Alright, so this is the reason why we use PCR. Now coming back to the three steps that are the major steps of PCR. Number one is step. Okay. So the three steps, the very first step is denaturation. The very first step is denaturation, wherein the two strands of the DNA are being separated. And how they are separated? With the help of a very high temperature. So I'm going to discuss each of the steps in detail, but let us first have an idea. So this is the first step. Step number one is denaturation, which takes place at a very high temperature. Next step, step 2 is annealing. Now what happens in annealing? When we have two different strands of the DNA, we need to put one primer, one RNA primer on both the strands, right? So here two RNA primers, one on each strand is being attached to the and then the 3' OH group is being left so that more nucleotides can come in and join. Now this particular step occurs at a different temperature than the temperature that is being used for the first step, that is denaturation. Then after annealing, the third and the last step is extension. The third and the last step is extension. Now extension means to extend something, to add up. Now in this particular step, what happens is that various nucleotides, they come up and they make a chain. They gets added to the pre-existing strand where the primer is being attached. And finally, after this step, when the extension is finished, we have two different DNA molecules. So initially when we started, we had one single DNA molecule, and from those one single DNA molecule, from that molecule, now we have two separate DNA molecules. And now we can repeat this particular cycle a number of times, so as to get more number of DNA copies. So now let us quickly discuss these steps one by one. Let's start with denaturation. Now this is the first step that we are going to talk here that is denaturation. Now these are the two strands of the DNA molecule. As we can see that these two strands are anti-parallel. Now this is the 3' end of first strand and this is the 5' end of the first strand. Talking about the second strand, this one is the 3' end and this one is the 5' strand. Okay. So now both of these strands are together. Our purpose in first step is to denature them, to separate them. And how we are going to do that? We can use a dilute acid. Supposedly I can use dilute HCl, okay, or I can use a dilute base, or I can also use a very high temperature. Yeah, mostly the treatment of choice is high temperature. So I am going to employ here a temperature between 90 degrees Celsius to 94 degrees Celsius. Preferably I am going to use 92 degrees Celsius, but the application of this particular temperature should be very short duration, it should be for a very short duration. Why? Because we are only concerned with the breaking of hydrogen bonds of these two strands. We are not focused on breaking the phosphodiester bond. We don't want that. Yeah. So for that, the employment of high temperature should be of very short duration. It should not be for the long duration. Otherwise, the phosphodiester bonds may get broken. So now the time duration is very less. So the time duration is very less, and now after this employment of high temperature, we have got two separate strands. Now the first strand again says that this one is the 3' end and this one is the 5' end. and that goes opposite for the second strand. This one is the 3 prime strand, and this one is the 5 prime strand. So after the denaturation step, what am I left with? What did I get? I get 2 different separate strands, and now I can use each strand to have another strand, and then I can simply amplify my DNA. Yeah, so this is about the first step, that is denaturation, where the bonds between the 2 strands of the DNA, the hydrogen bonds, are being broken apart and we get 2 separate strands. After this step, we will move to the annealing step. That was the second step, or we can also say that is the primer annealing. Let's go. Alright, so the second step is primer annealing. So now, because we have these 2 strands of DNA separated in the denaturation step, now we are going to attach the primers here. Now, what is a primer? A primer is an oligonucleotide of RNA that on which the new strand of DNA can be synthesized, right? So now this is the first strand of DNA. So now the question comes that where am I going to attach the primer, or the primer is going to be attached at what place? So now say this is the RNA primer. So because we have the 2 strands should always be anti-parallel, if this is 3 prime, then this end of primer is 5 prime and this end is 3 prime. Now this end of the primer has OH group attached, that is the functional group. And now from here a new nucleotide can come and attach to the OH and the RNA primer. Okay, so now whatever molecule we are synthesizing, it grows towards the 3 prime end. So it will move from 5 prime towards the 3 prime end. So now new nucleotides will come here and get attached towards the 3 prime end of the primer. So now because a new strand will always grow towards the 3 prime end, that is the reason that the primer will always come to the 3 prime end of the parent molecule. Similarly, when we consider this particular strand, in this case also, the RNA primer will attach to the 3 prime end of the parent molecule. Now when this parent molecule has its 3 prime end over here, then this RNA primer will have its 5 prime end over here, so that the 2 strands will be anti-parallel. Now this is again the 3 prime end, and this third carbon has the OH group attached here. So now whatever new nucleotides will be coming, they will be coming here and they will get attached to this particular OH functional group. So these are the 2 RNA primers that are going to be attached at the 3 prime end of the parent molecule. Now there are certain different things that we need in our mixture so that the nucleotides can come here and get attached to form a new DNA strand. Let us see what are these materials. Okay. So now what are the things that we require for the primer annealing? Number one thing which is very important, very evident, is the primer itself. So we need RNA nucleotides, we need RNA primer to be attached to the strands of the DNA. Why? Because we want to synthesize more DNA strands. So for the synthesis of new DNA strands, unless and until we won't have the primer, we are not able to synthesize new DNA. So apart from this primers, we need certain other enzymes that are going to help the complete process. Yeah. So now this particular step of primer annealing is also called as hybridization. It is also called as, where should I write? Yeah. It is also called as hybridization. Why? Because there is DNA RNA hybrid formed. Yeah. So because of the formation of DNA and RNA hybrid that is being formed during this step, it is called as hybridization. This process, this step is particularly called as hybridization. Now this is the RNA and this black color is the DNA. This DNA hybridizes with this particular RNA, hence the name hybridization, right? Moving on to the, before moving, let me quickly tell you that this particular step operates at a different temperature than the denaturation. So now the optimal temperature for this particular step is somewhere between 50 degrees Celsius to 60 degrees Celsius. As compared to the denaturation step, you must be aware that the denaturation steps occurs at a temperature of about 90 to 94 degrees Celsius, whereas this particular temperature, annealing, it occurs between 50 degrees Celsius to 60 degrees Celsius. So now when we talk about PCR, the employment of the temperature and the variation in the temperature plays a huge role in the formation of our products. Okay, let's move on to the next step, that is extension. So now let's talk about the last step of PCR, that is extension. And extension means when you are extending something, So now why do we say that this is an extension process? Because here the nucleotides are being added for the formation of the new DNA strand. Okay, so in the previous step, when we were talking about hybridization or primer annealing, the primers have already been attached to the separated DNA strands. Now this parent DNA strand, it has 3' end over here, and this RNA primer has 5' end over here. Now this is the 3' end upon which new nucleotides will be added in the step that is called as extension. Similarly, this is the other separated strand and its 3' end is here. And look at here carefully, this RNA primer has 5' end over here and 3' end on this side. So whatever nucleotides will be getting added will be added onto the 3' end, and this is how it is going to extend in this direction, right? There are certain necessary things that are to be added here. Number one thing is DNA nucleotides, okay? DNA nucleotides, and also it needs enzyme. Now another important thing before discussing these two things is the temperature requirement. Now this is the third step, and now the third temperature is being required. This particular step occurs at a slightly higher temperature, and the temperature is 72 degrees Celsius. So now what happens in our body when our DNA polymerase works at a temperature of 37 degrees Celsius? But consider when we are applying this particular step of extension, the temperature here is 72 degrees Celsius. So is our DNA polymerase going to be effective at this particular temperature? No, absolutely not, because the enzymes are not active at such a higher temperature. So at this high temperature, we need a thermostable enzyme. Now the question comes, where are we going to find that particular thermostable enzyme? The answer is very simple. There is an archaebacteria, that is Thermus aquaticus, and this bacteria aquaticus, this particular bacteria has thermostable, it's a thermophilic bacteria that is able to survive at a very high temperature, higher than 100 degrees Celsius. And this particular Thermus aquaticus bacteria synthesizes a DNA polymerase that is thermostable, that is, it is stable at a very high temperature, And the name of this particular DNA polymerase enzyme is Taq polymerase. T stands for thermus and AC stands for aquaticus. Taq polymerase enzyme, and this is the enzyme which is often being asked in most of the competitive exams, in your NEET exams, in your other competitive exams, in CSIR net exams, that what is the name of the DNA polymerase enzyme which is used in the PCR. So you have to simply mention that Taq polymerase is an enzyme and it is being isolated from a thermophilic archaeobacteria, that is Thermus aquaticus, which is actually efficient at 72 degrees Celsius. So now when we have this enzyme and these dinucleotides with us, what happens is then that now these nucleotides will come and attach to the 3' end of this particular RNA primer, and this is how the polymerase chain grows. The new DNA chain grows. Okay, and now similarly for this strand also new nucleotides will be coming, and this Taq polymerase will be able to attach these particular new nucleotides and synthesizes the new strands of the DNA towards the 3' end. So now this is how the new DNA strand grows towards the 3' end, and here also, because this is the 5' end, and now this will be the 3' end. So now initially when we started, we had one single copy of a DNA, then we separated that copy of DNA into two strands, and at the end of the extension step, now we have two separate molecules of the DNA. Now these three steps, number one, denaturation, number two, primary annealing or hybridization, and number three is extension. This particular cycle, this is one cycle, right? So from one cycle, one DNA is being converted into two DNAs. Yeah, we are getting two copies of the DNA. Now, similarly, we can repeat nearly 35 cycles or maybe more. So imagine how many copies of DNAs can I make? Isn't it great? Isn't it like some miracle that from one single DNA we are able to make billions of copies of DNA. So we are going to discuss the advantages, disadvantages and different types of PCR in the upcoming videos. Stay tuned for the next videos. Let me know in the comment section, did you like the video? How do you find it? Please comment, please like this video with your share and friends. Don't forget to subscribe the channel. Until next video, goodbye.